Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis.
Osmosis and tonicity are often difficult concepts for introductory biology students to grasp. Simply put, osmosis is the diffusion of water across a membrane from an area of higher concentration to an area of lower concentration. Tonicity represents the relative measure of dissolved particles or solutes in a solution. It is important to recognize that the higher the tonicity, the greater the concentration of solutes in the solution, and therefore the lower the concentration of water. Thus, a solution with 25% sugar contains 75% water. Here, we see a common lab experiment in which a bag containing 25% sugar is placed in a solution of pure or 100% water. The contents of the beaker have a lower solute concentration and higher concentration of water than the contents within the bag. Over time, water diffuses from the higher concentration in the beaker to the lower concentration inside the bag, causing the bag to swell. In general, water diffuses away from hypotonic solutions. Therefore, we can say that the beaker is hypotonic, which means lower solute in relation to the bag. In this experiment, water may move across the membrane of the bag, but not the sugar. In the next experiment, the solute concentrations of the beaker and the interior of the bag are the same. The term for this condition is isotonic. Iso means the same, and since these solutions are the same, over time we do not expect any net movement of water, and the bag should remain approximately the same size and weight. In the final experiment, watch what happens when a bag containing 25% sugar is placed in a beaker containing 50% sugar. In this case, the solute concentration of the beaker is higher than that of the bag. Since the concentration of water in the bag is now higher than that in the beaker, the water diffuses outward, causing the bag to shrink and lose weight. The term for this condition is hypertonic, with hyper meaning higher. There are some important conclusions you can learn from this experiment. First, notice what happened in both beakers A and C. In both cases, water moved from hypotonic to hypertonic solutions. The water, therefore, diffused toward areas of lower concentrations. Second, the terms hypertonic and hypotonic are always used in relation to one another. For example, in beaker C, we compare the hypotonic contents of the bag to the hypertonic contents of the beaker. Our bodies, and indeed all living organisms, regulate the movement of water by adjusting the solute concentrations in our cells. Therefore, an understanding of tonicity plays an important role in understanding how our cells and bodies function.
wants you to know about two extracellular components, and they specify one in a plant cell and one in an animal cell. So if we start with that in the plant cell, their example is the cellulose cell wall, which is found around the outside of the plant cell, outside of the cell membrane. But the cell membrane and the cellulose cell wall have distinctly different functions. The cell membrane controls what enters and exits the cell, whereas the cellulose cell wall maintains the cell structure and regulates its turgidity by resisting water uptake. Let's explain that. So, when a plant takes up water, the water is able to move through the cell membrane and into the plant cell's vacuole, and it increases the size of the vacuole. When the vacuole presses against the cell wall, so the cell wall is preventing excessive water uptake at this point, it's creating a hydrostatic pressure, the water pushing against the cell wall, and so we refer to this cell now as being turgid. And what happens then is that your plant is supported and stands upright, so you can see the little plant in the bottom right hand corner. However, if water leaves the cell, after the cell has become turgid, the water could possibly leave the cell. If you don't water a plant, for example, then it leaves. That hydrostatic pressure is decreased. At this point, the vacuole is not pressing against the cell wall, so we refer to this cell as being flaccid, and you can see that the plant at that point begins to wilt.
Plant cells are surrounded by strong cell walls, which can withstand high internal pressures without bursting. In a hypertonic solution, the cell membrane shrinks within the cell, but the cell wall remains intact. This is called plasmolysis.
The surface area of a root is enormously expanded by thousands of root hairs and symbiotic mycorrhizae. The root hairs are in direct contact with water and dissolved inorganic ions held in tiny spaces between soil particles. Water and ions can travel into the root by the apoplastic route, moving between cells and along cell walls, or by the symplastic route, moving from cell to cell through plasmodesmata. A molecule or ion may also move through the root via a combination of these two routes, or by moving slowly from cell to cell across plasma membranes and cell walls. How do water and minerals get to the leaves? Although roots can exert a slight upward pressure, it is evaporation of water from leaves, a process called transpiration, that moves water molecules and ions up from the roots. Transpiration exerts a pull that is relayed downward along a string of water molecules from leaf to root. Hydrogen bonds cause water molecules to stick together, a phenomenon called cohesion. As each water molecule evaporates, it pulls on the next water molecule and it pulls on the next. This relays the pull of evaporating water molecules all the way down to the roots. The adhesion of water to walls of the xylem cells helps to keep gravity from pulling the water molecules back down. As each water molecule escapes from the leaf, it pulls a column of water molecules upward. Solutes are transported along with the water. Thus, the plant xylem uses the movement of evaporating water molecules, cohesion, and adhesion to draw water and dissolved minerals from the soil into its roots and upward to its leaves. Hot, dry, windy conditions increase transpiration. If not enough water moves up from the soil to replace the water lost to evaporation, the plant will wilt and it could die. How does a plant prevent excessive water loss? An opening in a leaf is called a stoma. Stomata are generally open during the day to allow carbon dioxide into the leaf, where it is used in photosynthesis. While the stomata are open, water moves out of the leaf by transpiration. This allows the plant to move water and minerals to the leaf, and the evaporation of water also functions to cool the plant. If hot, dry conditions require the plant to conserve water, guard cells close the stomata. This is a trade-off. Although closing the stomata reduces water loss, this also slows down photosynthesis and may cause the plant to overheat.